Hello everyone, my name is Suwa Lee. Today, I'll be discussing effects of dietary protein concentration on digestibility of amino acids in diets fed to growing pigs. This work was presented in 2024 ASAS Midwest meeting. To avoid the impact of microbial fermentation in the hindgut of pigs, and because almost all amino acids are absorbed in the small intestines, digestibility of amino acids has been determined at the end of ileum. In addition, by correcting the apparent ileal digestibility, AID, for the basal endogenous losses of amino acids, standardized ileal digestibility, SID values can be calculated, and these values are believed to be additive in mixed diets fed to pigs. One of the reasons value for the SID of amino acids are additive is that the values are not affected by levels of amino acids in the diet. However, when we evaluate feeding gradients, sometimes we use diets containing a wide range of dietary protein, and it has not been known if levels of protein or amino acids actually affects the SID of amino acids, even though that's what we have learned in the textbook. Therefore, we hypothesize that the SID of amino acids would not be influenced by dietary protein, even if dietary amino acids are below, at, or well above the requirements of pigs. To test the hypothesis, 4 series of experiments were conducted. Data from the first 2 experiments were already presented in the 2020 Midwest meeting. However, to help you paint a bigger picture, I'm going to present all data from the 4 experiments today. The initial body weight of pigs used in all experiments was approximately 20 to 24 kilograms. In the first experiment, soybean was used as the sole source of amino acids in diets. There were 4 diets containing 4 levels of soybean meal, from 10 to 70% to create 5 to 33% crude protein in diets. A nitrogen-free diet was prepared to determine the basal endogenous losses of amino acids. Linear and quadratic effects of increasing soybean meal were determined in this experiment. Before we further discuss the SID data, let me show you data for the AID, which may be interesting for you. Data from all 4 experiments indicated that there were quadratic increases in the AID of amino acids as dietary protein was increased. This was in agreement with what we have learned. Are you familiar with this figure? Yes, this figure is from the terminology paper published by Dr. Stein and his colleague in 2007, and the AID data from all experiments showed similar pattern, indicating that as dietary amino acid increased, the AID values represented in orange went up and the increases slowed down. This is because the proportion of endogenous amino acids in ileal digesta is greater when dietary protein level is low. However, the proportion of endogenous amino acids is reduced as dietary protein increases, which results in an increase in the calculated value for the AID. Jumping to the SID results, let me set up the slide first. The horizontal axis represents the inclusion rates of soybean meal in diets, 10, 30, 50, and 70%. The vertical axis represents SID of amino acids in soybean meal in percent. Results from the first experiment demonstrated that as soybean meal in diets or dietary protein levels increased, the SID of arginine, isoleucine, methionine, tryptophan, and valine was not affected. This was also what we expected because we have learned that values for the SID of amino acids are not affected by dietary protein levels. However, the SID of histidine, leucine, lysine, phenylalanine, and threonine present here was reduced or tended to be reduced by increasing soybean meal in diets. This is because when we increase soybean meal levels in the diet, not only amino acids are increased, but also other nutrients are increased. For example, oligosaccharides such as stachyose and raffinose levels are also increased by inclusion rates of soybean meal. In this experiment, the analyzed stachyose and raffinose were approximately 5 and 1.3% respectively. Therefore, it is possible that increasing soybean meal in diets increased the level of stachyose and raffinose in the diet, which reduced the SID of amino acids. To eliminate the effects of oligosaccharides from soybean meal, experiment 2 was conducted with soy protein concentrate because to produce soy protein concentrate, the low molecular weight sugars are removed by alcohol extraction. Compared with oligosaccharides in soybean meal, the analyzed oligosaccharides in soy protein concentrate were much lower, and the analyzed stachyls and raffinose in soy protein concentrate were approximately 3 and 0.4% respectively. However, the analyzed trypsin inhibitor in both soybean meal and soy protein concentrate was around 3 units per milligram, which indicates that trypsin inhibitor was not removed when soy protein concentrate was produced. I will get back to this later. Let's move on to experiment 2. A total of 6 diets were prepared. First 5 diets included 10 to 50% of soy protein concentrate to create 7 to 33% dietary protein, and a nitrogen-free diet was also used. The linear and quadratic effects of increasing soy protein concentrate in diets were determined. Moving on to the results, the horizontal axis represents the inclusion rate of soy protein concentrate in diets, and the vertical axis represents the SID of amino acid in percent. The results showed that values for the SID of arginine, isoleucine, leucine, methionine, phenylalanine, tryptophan, and valine were not affected by dietary protein levels if amino acids were provided by soy protein concentrate. And we can see that there were more amino acids that were not affected by dietary protein level in experiment 2 compared with experiment 1. And this can be explained by reduced oligosaccharides in soy protein concentrate compared with soybean meal. However, there were a few amino acids that were affected by increasing soy protein concentrate in diets. The SID of histidine, lysine, and threonine quadratically or linearly decreased as protein levels in diets increased. Results from both experiment 1 and 2 indicated that the SID of some amino acids is affected by increasing soybean meal or soy protein concentrate in diets fed to growing pigs. The reason the SID values were reduced may be because the capacity of pigs for absorption of peptides and amino acids into the enterocytes has a limitation, or because there are still anti-nutritional factors present in soy protein concentrate such as trypsin inhibitor. Again, the analyzed trypsin inhibitor in soy protein concentrate that we used in this experiment was similar with the analyzed trypsin inhibitor in soybean meal. Then, what if we use soy protein isolate, which contains almost zero oligosaccharides and is very low in trypsin inhibitor? In experiment 3, we prepared 5 diets containing 5 different levels of soy protein isolate to create 6 all the way up to 30% crude protein. An N-free diet was also used. The analyzed stachyls and raffinose in soy protein isolate used in this experiment were close to zero, and the analyzed trypsin inhibitor was below the detection limit. You can see that most anti-nutritional factors from soybean meal and soy protein concentrate have been eliminated in soy protein isolate. Results from experiment 3 indicated that among 10 indispensable amino acids, the SID of histidine, leucine, lysine, methionine, and tryptophan was not affected by dietary protein if protein was provided from soy protein isolate. However, there still were some amino acids that were affected by increasing dietary protein, even though the difference among the diets were not too big. Again, it is possible that there is a reduction in the capacity of pigs to digest or absorb amino acids if dietary protein is well over the requirements for pigs, or a few anti-nutritional factors still exist in soy protein isolate. Therefore, we conducted an experiment where casein was used because casein does not contain most anti-nutritional factors that soy products have. In experiment 4, we used 5 diets containing 5 different levels of casein to create 6 to 29% crude protein in diets, and an N-free diet was also used. Results indicate that the SID of all indispensable amino acids was not affected by dietary protein. In the first experiment, we started with soybean meal, which contains soy-related anti-nutritional factors including oligosaccharides, fiber, trypsin inhibitor, and so on. Therefore, the effects of oligosaccharides were eliminated in the second experiment by using soy protein concentrate. The next experiment was conducted with soy protein isolate, which did not contain any oligosaccharides and lower trypsin inhibitor. However, the results from the 3 experiments indicated that the reason the SID of amino acid is changed by increasing soy products is that these feed ingredients contain fibrous components in addition to trypsin inhibitors and unique protein structure that hinders the amino acid absorption, even though soy protein isolate contains very low anti-nutrient factors. Moving on to the conclusions of this work, the AID of amino acid was increased by increasing dietary protein, and this applies to all feed ingredients that we used. This increase in the AID of amino acid can be explained by different proportions of endogenous amino acids in the ileal digesta when protein level was increasing from low to high, which results in an increase in the AID of amino acids. Results from experiments 1, 2, and 3 indicated that the SID of some amino acid in soybean meal, soy protein concentrate, and soy protein isolate was not affected by increasing dietary protein from around 5 to 3%, meaning that pigs appear to have a high capacity for digesting peptides or amino acids. However, there were other amino acids that had reduced SID when the soy proteins were increased in diet. I have listed the indispensable amino acids here that were affected by dietary protein in this experiment. Please be aware that the SID of these amino acids is reduced when dietary protein is too high. In addition, I would like to point out that threonine was mostly affected by increasing the inclusion rate of 3 soy proteins, and from this result, we can tell that the reductions may be related to increased endogenous losses because threonine is the most abundant amino acid in mucin proteins. However, when casein was used in diet, the SID of most amino acids was not influenced by dietary protein. The implication of this is that the direct procedure may be used to determine the SID of amino acids in a wide range of feed ingredients if dietary protein is not too high. Therefore, values for the SID of amino acids in feed ingredients may be determined below, at, or slightly above requirements without impacting results, even though the feed ingredients contain some anti-nutrient factors. Thank you for listening to my presentation, and I would like to acknowledge everyone from Dr. Stein's lab. If you want to learn more about our research that we are conducting in the Stein Monogastric Nutrition Laboratory, please visit our website. See you next time.